This is the first preparation for lecture 24. So we're starting in on chapter 5. We're going to look at the body plan in the chicken mouse. This time, next lecture, we'll talk more about somites and neural crest and anterior-posterior patterning. So we'll take probably about 3 lectures to get through this section. And just a point, which I'm sure you already know, but we're talking about amniotes. The chicken, the mouse are amniotes because their embryo develops inside the amniotic sac that has the fluid in it. And so some of the way organisms are categorized, as you'll know if you've taken evolution, is there are certain developmental features that are part of how organisms are classified. So bilateria, organisms with a bilateral symmetry; amniotes, organisms whose embryos develop in an amniotic sac to protect from desiccation and to cushion for protection. So we're going to be looking at, this is a mouse embryo. Don't worry about gene names or anything. But the question is just how, in amniotes, how is there an anterior-posterior axis formed? How is the brain and the spinal cord formed following gastrulation? We have talked a little bit about the neurulation process in Xenopus, how neural ectoderm is specified by lifting BMP inhibition. And we'll talk more about, in this section, about the actual sort of neural tube formation and that sort of thing. And then how do we produce some of the non-neural organs along that anterior axis? So you've got along the dorsal side your nerve cord, and that's going to form vertebrae around it for protection. But then you're going to have structures forming to either side of that, the paraxial mesoderm. So paraxial beside the axis, and axial mesoderm is the notochord. Axial meaning along that anterior posterior axis. So this is just an overview, and we'll just be looking mostly at this part today. And I'm going to do 2 separate recordings, one for the chick part and one for the mouse. Okay, so the initial anterior patterning in the chick embryo, you will remember that there is the posterior marginal zone, and here it's not very visible. There's nothing much to see visibly, but the posterior marginal zone is a group of cells that is going to form a thickened region, which leads to the formation of Kohler sickle, which is this sort of crescent moon shaped set of cells at the posterior end. And this is where gastrulation is going to begin, and the primitive streak is going to form. And remember that in that groove you have cells that are moving around, and then they're moving inside into the subgerminal space. So the cells are moving inside, and they're doing so not by involution, as happens in the frog and some other types of embryos, but through ingression. Okay. And the early chick embryo, it's hard to study because the development is taking place while the egg is still inside the hen, and in the very earliest stages there's not even an eggshell, and the egg white hasn't been laid down. So you know, by the time the egg has laid, there's the blastoderm that looks something like this, and it has, you know, tens of thousands of cells in it. So there is evidence from early manipulation experiments that show that the posterior marginal zone has the signals and information that initiate the formation of the primitive streak. And so you can take the posterior marginal zone cells from one embryo and transplant it onto an embryo that's approximately the same age, so its primitive streak hasn't started quite yet. And if you do that, you can get the formation of 2 primitive streaks, one that's the normal one and one that is the result of the transplantation of that posterior marginal zone. The reason why I've underlined sometimes here is because it depends greatly on the age. If the primitive streak had started to form already, then this transplanted cells will not induce one. Okay, so if they're not the same age, if you have an already existing primitive streak, there is a kind of inhibition of the formation of another one. And the signals that we're going to be talking about are FGF, TGF beta, so VEG1 is one of them, and then Wnt, and Wnt8c is one of the members. And these are part of the signaling that are required for formation of the primitive streak and then Hansen's node afterwards. So one thing that's really interesting is that as the egg is passing through the oviduct of the female, of the chicken, there is a position in the oviduct which is called the shell gland, and this is where the shell is being secreted and laid down around the egg. The egg is traveling in this direction with the narrow end first, so the narrow end is sort of downward, and the egg sort of rotates around and around and around. And as it's rotating in that way, it's rotating in this direction, and the yolk sort of stays in the same place. So the embryo itself doesn't, it's quite slow, right? It takes 6 minutes to turn once all the way around. So it's not rapid, and it's slow enough that the egg yolk with the embryo floating on the top mostly stays with the embryo sort of at the top. But because there is a little bit of this rotation in this direction, it is sitting on an angle. And so, and that the part that's uppermost, so the furthest from the source of gravity, this is going to be, excuse me, where the posterior marginal zone develops. That's going to be the posterior end. And it turns out that this also specifies ventral. So both the anterior, posterior, and dorsal ventral axes are defined. It's really interesting because in the chick there isn't very much maternal messenger RNAs and proteins and so on laid down in the embryo. A lot of it is set in place by by this process. And so after the egg is laid, this is not to scale. The blastoderm is still, relatively small, and so the egg has been turning in that direction. The blastoderm has been tilted, and so here's our posterior marginal zone. And it turns out that the very early part of the streak, the cells that internalize are destined to form ventral structures. And then as the streak moves more towards the anterior, at the anterior region, it's more of the dorsal mesodermal structure. So things will rearrange inside the embryo at some point to lay them out properly. But so basically, both axes, dorsal ventral and anterior posterior, are set up by this sort of interesting gravitational process, and Wnt8c, VEG1, and nodal expression are part of the signals that set up the organizing center. Now it turns out that at this stage, even at the stage of approximately 20,000 cells, you can take this before the primitive streak has started, and you can cut it into little pieces, and each of the little pieces will start forming their own primitive streak and setting up their own axes. So whatever signals have taken place, they happen before the primitive streak shows up, and they happen, they're so regulative that even when we have 20,000 cells, you can cut the embryo into smaller pieces and make a whole lot of little embryos that can still go through the process of gastrulation and forming a little tiny embryo. Okay, so now we're going to take a look in a little bit more detail. So this is Kohler sickle. So if you are looking down on the blastoderm, Kohler sickle is this little sort of, you know, crescent-shaped bunch of cells here. They look physically different from the others. And it's because they are actually, so if you were to slice this way and then look in that orientation, these are those cells. So they would extend towards you and away from you to form the sickle. And so the posterior marginal zone, which is this part here, these are a bunch of thickened cells, and they are starting to express the VEG1 and Wnt signals, and they signal to the cells of the epiblast right ahead of them to start expressing nodal. However, the hypoblast is still lining. So just go back to the early chick lecture. The hypoblast is a layer of cells that have pushed in from the embryo on the sides and are lining the bottom of the subgerminal space. And as long as those hypoblast cells are there, they are expressing Cerberus, and Cerberus is antagonizing the nodal function. Okay. Once cells have moved further from the embryo and have pushed the hypoblast cells out of the way and replaced with endoblast cells, now we don't have a source of that Cerberus anymore. So we've basically lifted the negative regulation. And at this point, Kohler sickle expresses FGF, fibroblast growth factor, which is a positive signal, and nodal signaling signals back on itself to activate its own expression. So we're maintaining the nodal signal with FGF, and nodal is maintaining its own signaling. And as it does this, it causes the nodal expression to move forward. And so that's the sort of the process of that. The nodal will keep regulating its own expression. It can be inhibited behind and then expressed here, and then inhibited behind and expressed here, and so on. So it kind of goes on like that. I'm going to stop here and we'll pick it up in the next one.